Yeah, well, thank you, Craig. I'm delighted to chat with all of you. I mean, this is a group of folks, I don't get, my sphere doesn't get much larger than the chemists and chemical engineers. So this is fantastic for me. Um, all right, let's share screens here. Now this is going to be pretty organic chemistry heavy. I mean, you did all invite an organic chemist. Okay, so here we go. Trying to get your faces a little bit over so that I can see my own slides here. Oh yeah, no, you're watching my screen, so I need to go on play. Okay, so, I think Craig described what we are doing pretty well. A lot of what I'm going to discuss is the use of what are known as reversible covalent bonds and how reversible covalent bonds could establish a genetic code maybe elsewhere in the universe, not a genetic code based on hydrogen bonding and hydrophobic effects like we have with nucleic acids. So I called this information storage and abiotic sequence-defined polymers. So polymers other than nucleic acids and their potential for replication and seeking molecular complexity. And this little slide here shows just one example. I'm just wanted to introduce with this. This is a reversible covalent bond. It's a disulfide linkage. And you may know in proteins, our protein structures are heavily controlled by the formation and destruction or breakage of disulfide linkages between our cysteines amino acids. And a lot of organic chemists have exploited this for a variety of purposes. We're going to talk about exploiting reversibility and kinetics of covalent bonding for these purposes of information storage, potential replicability, and that maybe life could be based on these kinds of interactions elsewhere. Now, I already alluded to a main comparison that I'm going to discuss is that with nucleic acids form the genetic code here on planet Earth. Um, we're probably pretty well versed in the fact that thymine pairs with adenine, cytosine pairs with guanine, and this ATCG interaction is the language, if I can use that term, because it'll help to start prepping us for where we're going. It's the language that has evolved for the nucleic acids to store the information that creates the machines that run life, where I discuss the machines being the proteins. So here is another potential language. Instead of ATCG, this is a set of 4 reversible covalent bonds. This top pair is known as a boronic acid with a diol, but you might want to just call it B and D instead of A and D, okay? And boronic acids with diols make these what are known as boronate esters. Another reaction is what's known as a conjugate addition. I mean, how many people here have had organic chemistry? Is is that something most everybody's taken or absolutely not?
I think I had a little as an undergraduate 50 years ago. That helps. Yeah.
One undergrad
It does not
class.
help. It does not help. No,
So a whole lot,
I'm with you, sort of.
yeah, so a whole lot of what would help me out is, when I get too organic heavy, just stop me. Okay. All right, but in the second pair, there's what's known as a thiol. It undergoes a reaction with this conjugate acceptor is the terminology to make these. Another is, this is called a hydrazine. It condenses with an aldehyde to make hydrazones. And finally, metal chelation with a variety of synthetic ligands, as implied here with purple, will make these kinds of pairings. And I refer to these as, orthogonal dynamic covalent reactions.
Eric,
Or
could you say what the importance of the reversibility is?
yeah,
We get into
jump,
that?
absolutely. I love it. So just like A and T are totally reversible, so that the genetic strands can dissociate from one another and therefore make a complementary copy, we would like these bonds to be reversible. And if you are imagining some life form based upon this, you don't want everything to just react and never reverse, because that would basically be a
Gotcha.
way of considering death.
Thanks.
Okay. Yeah, but those are the kinds of questions I really need. Thank you. Well, what I'm implying here is all four of these could be mixed, and they only make these pairs. And that's why we refer to them as orthogonal. And in fact, if you pair A and T and C and G together with no polyphosphate backbone, no ribose rings, they all interact with each other totally. So
Mm.
these pairings here actually have higher fidelity than our current genetic code does. Well, so can we make strands? I'm going to go through a lot of examples of little pieces of what I'm going to call the grand vision of writing in a completely abiotic fashion information. That's the big goal of this talk, probably. How do we write information in an entirely abiotic fashion where the concept is, can that be a basis for information that's being stored elsewhere in the universe?
Cool.
So what we've done here is taken some peptides where certain amino acids were ones that we synthesize, the X amino acids. The single letter codes are what biochemists, encode the amino acids of, where we put in these two X's from different lengths. Sometimes we'll put in four X groups, and the amino acids that we're referring to will be an aldehyde and a hydrazide, which was one of those four reversible covalent bonding pairs on the previous slide. And now I'm going to use a little cartoon to represent that molecule and this molecule. Well, if you start pairing them together, it's kind of like just the first step. Can you get structures that zipper up, much like nucleic acids do? If you put three aldehydes with three hydroxides, indeed, the only thing we make is that little zipper, and four of them, that's all you get. They very cleanly pair with one another, perfectly analogous to how a one complementary strand of DNA only pairs with its complement. We have 6 mers on 6 mers, 3 mers on 6 mers, and the cartoons here kind of say the whole story. And I'm not really taking you through the spectral evidence for the background of the concept. This is just another supporting slide of that concept. If you have a 6 mer and you add a 2 mer, or 2 equivalents of a 2 mer, 3 equivalents of a 2 mer, so on and so forth, we could follow that this makes very clean complementary duplexes. And of course, nucleic acid is called a duplex. Okay, we have made a pairing system that only pairs with its complement. That's kind of an upshot of this.
Eric, and the way you're doing these cartoons, it looks like you're getting the bonding down the middle and sort of a backbone-like thing on the top and the bottom. Am I over-interpreting what that
No,
cartoon represents?
you are not. So ultimately, the goal, and I'll show you some progress towards this goal, of the blue strand is acting as a template that is binding the complementary pieces
Yeah.
above that template that can then sew themselves up. I'm about to show you a little advanced in that regard, where the blue strand basically templated the synthesis of the red strand. This is exactly what nucleic acids do, of course.
Mhm.
Now, ultimately, the blue strand here would have different of the TORC pairs, or those reversible covalent bonding pairs. So instead of just going H, 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 okay, making A, you would have the language embodied in those reversible covalent bonding pairs being translated into the complementary pair.
Cool.
That's a major goal of what my group works on.
Just to follow that question, if I may, the backbone, how is that actually formed in
Yeah,
your?
my backbone here, all of a sudden my computer isn't letting me. I got to stop sharing. Can everybody hear me okay?
Yes.
Yes. And
Okay.
we see
Yeah,
your
my
camera.
computer wouldn't let me advance. So let's try it again. That may happen a time or two. So these complementary strands are made from peptides currently. So you could see my cursor because all of a sudden all
No,
your faces have vanished.
We're just seeing your camera.
Ah,
Just seeing
Jesus.
you.
You
Okay.
need to share screen again, I guess, Eric.
Yeah. Okay, I'm back. Now you can see it?
Yes.
Yeah. Yes, thanks.
Yes. I blundered. My fault. So to answer that question, the strands currently are peptides. So we've taken, a biotic polymer and inserted these little abiotic amino acids as a way to test this concept.
Thank you.
Mhm. Eric, we don't hear or see anything.
I think he may have frozen.
Yeah.
Oh, bummer.
That wouldn't be good.
I am having major problems, aren't I?
Yeah,
No, now
he's
you're back.
You froze.
Yeah, froze there temporarily.
Okay. Well, just bear with me while we keep trying this. Okay, I'm back.
You're back.
Let's hit play again. So to answer some of the questions, I already had alluded to that the ultimate goal is one template strand would carry a complement of the reversible covalent bonds, where we were trying to synthesize, replicate, if I can steal that term from biology, the complementary strand. And again, what this backbone is, that was alluded to in an earlier question, that's peptides currently, but I'm about to show you that towards the end of the talk that we're making our own artificial backbones also. Okay, so let's just see, is there any hope for this? Here's a peptide backbone that has two aldehydes, and we pair up the complement in blue, where again, my cartoon has a red and blue pair. And they again will anneal to the template strand, and then we use a synthetic step to sew up what would be the complementary strand. And indeed, that does actually happen. We put in just two of these, we link these together, and we make the complement. So this kind of cartoon down here is what we find. Now, it did do something that surprised us. It made a macrocycle instead of a linear strand, but I think we've sorted out why that is. So we can make a complementary duplex of just two monomers. Can we extend to 3 monomers and 4 monomers? Well, yes, we can. And it's incredibly clean. So if you take a template strand that has 3 aldehydes, add the hydrazide monomers, it nicely sews up into a 3mer. So we've taken the information of an AAA and been able to transcribe it into an HHH with essentially 100% fidelity. And also it can be done 4 times. Now, once again, it's okay for us that it made a macrocycle. We really didn't care, but that's not our ultimate goal. We'd really want this newly created strand to be entirely linear, not cyclic. And we're working on ways of fixing that. But the key upshot is we have this language of 4 complementary pairs of reversible covalent bonds. And at least in the context of one of those pairs, we can make a complementary copy. So if we're going to explore the chemistry such as that I'm going to go through throughout this talk, we got involved with how would you discover whether life elsewhere could do anything like this? How could you discover if life elsewhere is based on any kind of chemistry, not just nucleic acids? I'm going to make a statement here that I'd love to hear if there's any discussion about this, but I think it's nearly impossible to imagine that life elsewhere has evolved to use nucleic acids, proteins, maybe carbohydrates because they're so ubiquitous, but actually having A, T, C, and G attached to a ribose, attached to a diphosphoester as our information carrying, I think is kind of crazy. So when does chemistry pass a threshold to become biology?
There you go.
I'm very good friends with a professor at University of Glasgow, and I'm sorry, this is left over from a lecture we gave back to back several years ago. Um, his name is Lee Cronin, and he's come up with a molecular complexity scale that's referred to as the pathway assembly number. And the notion is that certain small molecule chemicals, well, yeah, that's chemistry. At some point, they start to assemble and make things that are complex. And ultimately, that complexity finally translates into biology. And where are these dotted lines on this scale? I don't know. He doesn't know. But the premise is that you're not going to look for nucleic acids. We're not going to look for proteins elsewhere. We have no idea what to really look for as far as the organic and inorganic chemistry that might generate life elsewhere. So instead, let's be totally agnostic and look for a molecular complexity, where the complexity of the molecules beyond a certain stage, they could only be generated by life.
Hmm.
This is an idea that he and I generated when we coincidentally ran into each other at a NASA think tank conference. And he has really taken the idea, moved it forward dramatically. So he has a molecular complexity scale that involves a graph theory mathematical analysis that assigns, here's a handful of molecules, assigns a certain level of complexity to those molecules. He's done this with thousands of molecules now, where simple carbohydrates or amino acids fall pretty low. Things like well, this is Viagra. I'm sorry, ATP, strychnine, certain things start to fall pretty high on this molecular complexity scale. And the concept is, let's use strychnine as an example. Would that molecule ever, under any circumstances, be created by happen chance chemistry? With a very specific stereochemistry, with very specific ring systems fused in a particular way with certain functional groups, it seems impossible, unfathomable that without a life form that would ever be created in any appreciable extent. Okay, so that's the molecular complexity concept. And he has a mass spec, I'm singing Lee Cronin's toutings here. He has a mass spec way to translate the fragmentation patterns of a variety of molecules and take them through some graph theory algorithms to assign what he calls their complexity number, 7, 16. And it seems that somewhere around, fuzzy around 15 or so in his complexity scale, well, he can pretty nicely start to separate away what are natural products found on Earth versus other small molecules that these down here we certainly know are created elsewhere in the universe, just entirely on their own by happen chance chemistry, like the Uri Geller experiments of so many decades ago. And things that are transitioning, these are might be biosignatures. And finally up here, you gotta say, wow, there has to be life.
Hmm.
And now what you would do, and I'm going to show you the grants for this, what you would do is put a mass spectrometer on a mission. And of course, NASA uses mass spectrometers continually. Sample the atmosphere, sample the planets, et cetera. Sample wherever you're going to be able to sample and run the mass spec fragmentation patterns and check to see if you get a molecular complexity level of anything that exists there that starts to exceed this threshold. That's kind of the life or biosignature concept. Well, we've teamed up with Andy Ellington. Andy's just a super close friend. And also again with Lee Cronin at the University of Glasgow to do this in a slightly different way here at UT Austin. And I'm not going to say I have a lot of results. I'm going to show you just what is the premise. So we think we could take mass spec fragmentation data coupled with proton 1 NMR spectroscopy data coupled with liquid chromatography MS, what's called LCMS data, a variety of very common spectroscopic methods that organic chemists use, and train some kind of machine learning algorithm on the spectroscopic data to generate a molecular complexity scale. And of course, contrast, it's all in collaboration with Lee Cronin, contrast to his molecular complexity scale. So my own group, for a variety of purposes, we use support vector machines, random forests, neural nets for a variety of different kinds of things in the group. So I'm throwing that premise out there that there's another alternative to maybe how to analyze the data that could come from a mission. Right now, we're already starting to process proton 1 NMR spectra. I mean, here's some score plots that you're probably used to looking at these kinds of things, of trying to group complexity of molecules across PCA axes as a way of starting to check to see, can we train some machine learning algorithm that would then check molecular complexity and assign it to some of these categories. The Ellington group has a totally different way of doing this. So I'm going to say this slide, this is what we're doing. Back here is what Lee Cronin is doing. Let me show you what Ellington is doing. Ellington's using something known as a proximity ligation assay using nucleic acids. Okay, and this I think.
Sorry, I have a
Oh, no.
question. Can
Oh
I?
yeah, jump in, please.
So, on polymers, how are polymers in your complexity? How would they be located in your complexity scale? If I repeat the same unit multiple times, do you consider this as a different complexity?
No.
So
Okay,
it's just the compound in the monomer itself.
It becomes a little more complex than the monomers themselves. Okay, the graph theory, because you're repeating a certain unit of graph theory over and over again, it doesn't add to the complexity. Now, and a follow-up to that question is, what about a random polymer of tons of different monomers? It would seem incredibly complex. Okay, well, now this is Lee Cronin. I'm trying to sing Lee Cronin's song here, not my own. It has to do with how much material is present. If you have a random polymer of, let's say, 10 monomers, you're not going to make much of any one of those polymers in any appreciable level. You're just going to have a gamish of tons of different random polymers. And that is not included in his complexity scale. So a key feature is you have to have a molecule of a certain molecular complexity in an appreciable amount. And of course, that's a gray area too. How do you define appreciable amount? Does that help? Okay.
Yeah, thank you.
Okay, so right here on our campus, what's Ellington doing? His idea is let's probe the complexity of a surface. How complex is a surface? Well, okay, let's just use an example of sand, silicon dioxide. The complexity of that actual exposed surface is pretty minimal. Let's another extreme. Let's look at the surface of a cell. Okay, it's filled with the glycocalyx. It's got proteins extruding. The proteins are dimerizing, trimerizing. They're moving around. The complexity of the surface is enormous. So as one potential life detection methodology is to analyze the complexity of surfaces, I'm talking 2D surfaces here. And the way Ellington does that is he probes the complexity of a surface by binding a nucleic acid strand. Let's make it crystal clear. The implication is not that there's any nucleic acid on that surface because we're not looking for nucleic acids. The nucleic acid is simply a binding unit that then binds, let's say, another position on that same surface with a different nucleic acid. And you do what's known as a ligation assay, where this nucleic acid is proximal to this nucleic acid, and you sew them up with a splint. This is kind of standard molecular biology. And then you characterize the nucleic acid sequences that arise. And you can characterize tens of thousands of them at a time with modern next-gen DNA sequencing techniques. Just like you could go get your own genome entirely sequenced right now if you wanted to spend the money, you can sequence all the nucleic acids that come out of this proximity ligation assay and probe their complexity. And now imagine you're trying to analyze sand. Well, it's pretty much the same nucleic acid kinds of sequences are going to bind the sand over and over and over again. So the complexity of the nucleic acids that come out of binding something as boring as sand is going to be really minimal. But the nucleic acids that bind a very complex surface, this will be different than this one, which will be different to another one, which will be different to another one across the surface. And they all bind together in proximity and you sew them up. And now the resulting nucleic acid that came out of probing the complexity of a surface itself becomes very complex. And that's the complexity scale that the Ellington group is trying to establish. This is all funded by a grant called Enfold. It comes from Sarah Johnson as the PI, the head PI at Georgetown University. Heather Graham is at the Goddard Space Flight Center. I've toured one time the Goddard Space Flight Center. I loved it. It was amazing. And we get sent fossils and biofilms and soil standards and we get sent all of this stuff routinely to do our analyses on these NASA-based samples. All right. I'm going to switch gears. So I've covered how we're trying to make a new language out of the reversible covalent bonding pairs that are orthogonal and hence can communicate and potentially and generate life elsewhere. I've now taken you through some projects of how to register biosignatures based on molecular complexity. I'm now going to take you through a different project that's really focused on information storage. How can abiotic polymers just like nucleic acids and peptides, but they're biotic. How can abiotic polymers translate information, store information, and translate that information? So in reality, although this is all stemmed and inspired from trying to understand how life elsewhere may evolve, I'm also going to show you that we're actually truly using this methodology to store information in a highly dense, compact form here on planet Earth. So I'm going to go through, this is the last topic, what are known as sequence-defined polyurethanes. You know, if you go down to Home Depot and walk down the chemical aisle, you're going to find varnishes made of polyurethanes. Lots of plastics are made of polyurethanes. What we're doing is making polyurethanes where each side chain, each R group, is entirely different from one another. And what that R group is encodes information. And if I can make the analogy in nucleic acids, you have A, T, C, and G, and those side chains of the nucleic acids encode the information that, of course, generates life. Can we encode information in plastic? That's kind of the big scale question here. So can I make, let's just start with the simplest question. Can I make a polyurethane that's sequence defined, just like is a peptide or a protein? Well, here's a short polyurethane with a different side chain here, a different side chain there, here, there, here. You can see that's a single molecule, and I'm going to show you Heptamers in a moment, where each side chain is defined. And if I make an encoding system where this side chain communicates a particular piece of information different than this side chain, different than this one and so forth, I've written a language into this polymer. Now I'm going to say something that's like painfully obvious, but it really helps to set the stage. Right here, I've written Rogelio Escamilla, the grad student who worked on this. And if I can be really pedantic about it, I've written Rogelio Escamilla with 26 different monomers. Some are lowercase, some are uppercase, but 26 different monomers in a polymer. This is a polymer of letters that we use to communicate a language. This is absolutely no different. This is a polymer of side chains that can be used to communicate a language. And I'll say it for the umpteenth time, just like DNA is a polymer A, T, C, and G used to communicate a language. If I can write the language, can I read the language? Okay, this is the methodology we developed that allows you to read out what is the information you've encoded. That's called LCMS. Not that it matters too much, liquid chromatography, mass spectroscopy, where we can take off one letter at a time from the end group. Let me say one piece of information. It's probably not good to call it a letter. One piece of information from the O terminal, one at a time. So it'd be like taking this word on the very bottom, oxazolidinones, and finding out what it says by removing the S, then removing the E, and then the N, and then the O, etc. And it allows you to read oxazolidinone. Here's some of the data. It's beautifully clean, gorgeous data. This is kinetics. Probably most people don't care. Okay, so can we do this on really large scale? I can tell you right now, we write 400 of these at a time with an automated instrument in my lab, where we can write 400 pieces of information or different oligourethane strands all in parallel. This is an old slide. We're up to 97, 98% yield in making a 10-mer, not even a 7-mer. We can make this 10 long and in an automated fashion get them at 98% yield, 400 of them at a time. That's the scale we're starting to write. Okay, yeah, more kinetic data. So let me give some tribute to a gentleman in France, Jean-Francois Lutz, who has really pioneered the area of using abiotic sequence-defined polymers as a molecular storage data methodology. So I'm moving it a little bit far from life detection and abiotic sequence replicators and simply asking, can we store information? And I'm going to say Jean-Francois really gave this field its birth. So I'm going to write Hello World. Now, I didn't want to write this. I'm having fun here. My graduate students convinced me I had to write this because that's what all nascent programmers supposedly first write. You can all confirm if that's true or not. Okay. And we took Hello World and did what's called a Huffman tree data condensing, where you look at the most common letter and assign it a binary code. And as the letter becomes more infrequent, it gets a more complex binary code. So in Hello World, it's pretty obvious L is the most common, so it's going to be encoded with a 0, 1, and you get this Huffman tree that will take Hello World and convert it to binary. So this is not the standard binary ASCII table where you need 8 zeros and 1s to encode every letter. This is its own special encoding system that does a data reduction. But I've got I've got a series of 0s and 1s. Well, to me, that's just because that's the way computers have to operate. I can do something a hell of a lot better than a bunch of 0s and 1s. DNA already does better because it's got A, T, C, and G. I can put any monomers I want in the backbone. And this shows that we take that binary string and we convert it to octal. Octal breaks the binary string into this number of units, and this is the octal code, which of course would be from 0 up to 7. These octal codes, all we have to do is make an encoding system where they're assigned to a particular monomer. So for example, 5745124 becomes 5745124, where the side chain is assigned to a certain number in the octal code. Okay, I think it's probably straightforward. I love this field. I've only been working in this for about a year, and it's so brand new that anything we do is novel. Nobody's ever done this. Nobody's ever tried to encode information in a piece of plastic. People encode in DNA all the time, don't get me wrong. It's encoding that's plastic that is new. So this was the abstract for the talk. Here we're going to sequence this oligomer. Here's the data that sequences it. This is just showing the organic chemistry and the actual data that we generate. And now if you take the masses of those oligomers as we sequence them, convert those masses back into the octal code, because every mass here stands for a certain octal code. You could take the octal string, convert it back into the binary string, and lo and behold, Hello World comes out. So we write a software Python script that analyzes all the data and simply spits out the phrase Hello World. Well, my goal is to write with 64 characters. I'm not there yet, so I'm going to show you 16. This is called a hexadecimal code. Here are 16 different monomers that are all mass resolved. So I'm a huge Jane Austen fan. I'm almost done with the talk, just to let you know. I'm a huge Jane Austen fan. You know, we wrote something out of the book Mansfield Park on marriage advice. Okay, just for fun. But if you see in the background, because this slide says it all, even though it's kind of a small slide, the background in red are all the oligomers that we wrote in one single shot.
Hmm.
Each single side chain is assigned to a hexadecimal code. Now, hexadecimal codes, if you look up an ASCII table, are combinations of numbers and letters. The hexadecimal code is converted back into binary through the Huffman data reduction tree, which then spits out the sentence or the phrase in English. And if one scheme of happiness fails, comma, human nature turns into another, you could see I'm reading the top. It doesn't matter. It's all automated. We write the polymers in parallel, we sequence them in parallel, and we run our software script, and it spits out what is the final piece of information.
Oh.
Okay, so what's the upshot of what I tried to show you today? We're nipping at the edges of many different things that we think could be involved with life forms elsewhere. We think it's nearly impossible to imagine that A, T, C, and G is how life is based elsewhere. So what other kinds of pairings in duplex formation could encode information? And we use these we use these reversible covalent bonds. We've shown that the reversible covalent bonds do allow information transfer, meaning one sequence will properly pair with another complementary sequence. And I even showed you how we're able to actually translate one sequence into the other sequence as a step towards replication. I'm sorry, that's what I'm showing down here. And now, can we store information in things other than alphabetic languages or ideographic languages other than DNA? Yeah, we could store information in plastics, is kind of the last topic that I discussed. So the summary here is, what chemistry are we going to find elsewhere? That's the big question. What are the tools we could generate to probe that chemistry? And can we show footholds in the fact that chemistry other than nucleic acids can store information, can replicate, and can pass that information on to future generations? That's kind of the big upshot of all the little things we're doing. So thank you. Thanks
Great.
for having me.
Oh, Eric, that was, I think,
Excellent.
just what I was personally looking for to understand what you're doing. I know there's a lot of depth under that. You've given us the tip of the iceberg, but that was a great summary. Now, I'd like to start off with a really, truly naive question. You do all this by squirting stuff into test tubes? Is that how you do it?
Let me see.
Well, you got gene sequencers and things, so it's more than that. But so I think you're talking about the technology behind it's probably a whole talk. I'm looking for something a little simpler than that.
Yeah.
But,
I got to, because my escape button won't I got to stop sharing for a second.
okay.
Okay, so the synthesis is all done in robots. So the robots have their own little tiny test tubes. That's certainly true. It is test tubes, but when can you see my fingers? They're test tubes about this
Yeah.
big.
Got it.
Okay, and they're all put into a arrayed in a sequence, and the robot comes in with little pins and reagents and delivers things to the various spots.
Got it.
This sequencing, like I implied in this slide, This is done on an automated instrument also. Now, this is a big instrument. It's a mass spec instrument, $150,000 instrument, but it's got a sample changer where, again, all the little test tubes are lined up and a little pin comes one at a time and samples each one and gives you that data readout.
Wow. Cool. So we have a truly intelligent question then? Yeah, Sean.
But I can't say it's necessarily intelligent, but I'm curious if we go with the supposition for a moment that it's not, that life might have a different basis beyond DNA elsewhere, then are some of our assumptions about the signatures, for instance, I'm thinking in particular the atmospheric ones, looking for disequilibrium chemistry, are we potentially looking for the wrong sorts of disequilibrium chemistry if the whole basis of life in a different system was, say, some of these polymer bases instead?
Okay, really good question. And I probably need to know more about what you're doing to find the disequilibrium question to give a proper answer, but I'm going to throw an answer out there anyway. If the disequilibrium chemistry is looking for conditions that are relevant to how nucleic acids replicate on this on this planet, then I would say, yes, you could quite readily be missing chemistry that is totally different. So I'm going to take you up here in the grant I have from NASA. It's a separate grant. I discussed this grant on life detection. That was this application. But I have a separate grant from NASA to explore these reversible covalent bonding units. And these are very robust at much higher temperatures where nucleic acids would just totally dissociate and be completely useless to replicate and transfer information. These can actually still replicate and bond and be reversible at temperatures approaching boiling water.
But
So
we have archaea that can survive in boiling water, and it works fine with DNA.
It does, 100% right. Now, the strands are much more robust. They've evolved to take that temperature, of course.
Okay.
But my point being, Craig, I didn't mean that DNA can't do this. My point is there's other kinds of chemical pairings that are far more robust at the high temperature.
Got it.
Yeah.
No, I do understand. Yeah.
Okay.
Okay, that ties into the question that I had, and that is that all of our suppositions about where to look for life elsewhere have been tied around having liquid water available. And partly from, it's from two basic aspects. One is the mobility that provides. And the other is that at least from the perspective of the naive astronomer, the chemistry can proceed energetically in an energetically favorable mechanism at those temperatures. And now you're saying that, maybe we're not looking at the right temperatures. Maybe you can do things at different temperature ranges. So I guess a lot of these things don't necessarily need liquid water, do they? And, what is the temperature range at which this can proceed?
Well, so these pairs work great in DMSO. And DMSO is boiling, water is 100, DMSO is like 212 or something like this. So yes, if you had, okay, it sounds so silly when I start to say these things, but maybe you're all used to this. If you had oceans of DMSO, okay, you probably could have a much hotter planet that uses this kind of pairing or others that I have never even thought of. That's kind of the point of the molecular complexity scale.
So in other words, the classical habitable zone around other stars is really an artificial construct of our myopia as to where to look.
Write that down, Bill. That's great summary.
I'm not going to shoot holes in the theory that water is essential for life. I've heard this many times when I go to the NASA conferences. That could absolutely be true. I am pointing out that it's clear one can envision organic or inorganic chemistry-based life or replicators, let me leave it at replicators and not call it life, that are using interactions that survive in much hotter solvents.
So it's true that all the life we know is based on DNA and that whole system, but suppose there were life on this planet right now based on some scheme like you're talking about. Would we know about that? It's not ubiquitous, obviously, but maybe it's down there in the black smokers.
Actually, there is such a life form on our planet called prions, which replicate solely based on proteins. So basically these are the agents that cause Alzheimer in our
Yeah,
brain.
that's true.
And
And
mad
so-
cow.
Yeah.
Yeah.
So the concept that Eric is bringing up is very similar to what potentially prions are actually. Well, of course they do differently. They work different, but I would really recommend you guys to look up prions, which basically replicate without DNA essentially.
Interesting wrinkle.
Okay, I see we have a
Very
few
interesting.
hands up. I guess EL
I have
has a
to
question
tell
and
everybody
Rob
I have
has
a
one.
meeting at 2 o'clock. I'm really, really sorry. So, but
I
let's
don't-
go
I
all the
don't
way
see
till 2.
the hands. Bill, if you see the hands, I'd be happy. to-
Yeah, I see Rob has a question and EL has a question.
Yeah, I
So
like.
one of you.
Okay, I go first. So if you, what you have to fight against is always KBT, right? So whatever temperature you have in your environment, you have to make the bond stable, just barely stable enough so that they form and can be separated easily. If you go to higher temperature, right, like you do, then you get more stable reactions, and you can do this. There's no problem with it. Something that is a problem is that you need the stability of the backbone, and you cannot change the chemistry there. You would have to make it much more stable than your binding or bonding, the ones that carries a specific information. And if you're not able to make the backbone more stable, right, then you would lose actually this contrast. And what this means for evolution is that your molecules are not as stable in the environment as they are on Earth.
Well,
It's
hold
why-
on. In no way are these bonding pairs at all reaching the thermal chemistry that break standard covalent bonds. These they are covalent bonds. It's an excellent point. Their bond dissociation energies are upwards of 80 to 100 kilocalories per mole. So they're incredibly strong bonds. But so thermodynamically, they're very stable. But kinetically, there's mechanisms that allow them to exchange. So the backbones that we would use in the polyurethanes or the other kinds of replicators, those backbones are not going to break to your 300-400 degrees. Whereas these units are exchanging, and boiling water just fine, but the backbones are perfectly stable.
Interesting.
Yeah, but as closer as you raise the temperature, right, you get closer with the bond energies.
Well, you
in
certainly
order
do,
to-
but I don't know what solvents there are that aren't already boiling and gaseous at the point that but who knows what solvent might be out there in another planet. But most solvents that we're all used to have all boiled away before you're even breaking the bonds.
Okay, Rob, do you have a
But
quick
that makes an
question?
assumption you got to have a solvent. I don't want to make that assumption. I don't know.
Tricky business. Tricky business.
Okay,
Yeah,
Rob?
so I just have a clarification question. There's an implication here that there are many, many of these reversible covariant bonding pairs that one could devise, but you didn't actually say that. Is it true that in your estimation, there would be many, many of these bonding pairs that one could devise?
Absolutely, Edward. And our group works to devise new ones continually.
Mhm. And
Fascinating.
then one could choose these for the temperatures and bond and solvents as desired.
Yep, you probably took a sentence right out of my NASA grant. Absolutely right.
Thank you.
Cool.
I'm sorry, I have a group meeting at 2 o'clock.
Okay. Well, Eric, let me wipe up or end up with my my thanks. This kind of talk is exactly why I joined this group. so that we can have this sort of presentation and discussion. But just bang on. Thanks for
Agreed. This was fantastic.
Yeah.
Thank you.
Thank
Yeah,
you again.
Thank you so much. Okay.
Okay, thanks.